Good morning, students. This video is of biology for class 10 students. We'll be continuing the same chapter, absorption by roots, the process involved. Today, we'll start with the topic tonicity. Tonicity is the relative concentration of the solutions that determine the direction of diffusion. So, in relation with osmosis, we can define tonicity as the ability of an extracellular solution. Extracellular solution means the solution which is kept outside the cell to make water move into or out of a cell by osmosis. So, based on this, there are three types of solutions, namely isotonic, hypotonic, and hypertonic. Isotonic solution means iso means same, tonus means concentration. So in isotonic solution, the solution which surrounds the cell as well as the solution inside the cell sap are with same concentration. In hypotonic solution, hypo means lower. So here the solution which surrounds the cell is with lower solute concentration. That means the water molecules are more as compared to the cell sap. Then in hypertonic solution, hyper means higher. Here the solute concentration is more in the solution which surrounds the cell. So in all these cases, the cell membrane is separating these two solutions. Cell sap and the surrounding solution is separated by the cell membrane. So what would be the result if we place a plant cell and an animal cell in these three different solutions? So a plant cell, any plant cell and an animal cell, that is RBC, we place in these three different solutions: isotonic, hypertonic and hypotonic. So in isotonic solution, that means solution concentration is of same inside the cell and outside the cell, so there is no osmosis. In hypertonic solution, concentration solute concentration is more outside than the cell sap, so water move out or diffuse out, and this is known as exosmosis. And if it is placed in hypotonic solution, the endosmosis takes place. Water diffuse into the cell because here solute concentration is very less or water molecules are more. And in these three situations, in isotonic solution there is no change in the size of the cell. In hypertonic solution there is shrinkage of the cells takes place, and in hypotonic solution little enlargement of the cell takes place. And in plants, certain substances, certain nutrients are transported actively because the nutrients like the ions of nitrates, sulfates, potassium, zinc, manganese, etc., these substances are more inside the cell than the surrounding soil water. So normal diffusion is not possible here. So here these substances have to be carried forcibly inward or into the cell by the expenditure of energy, and this is known as active transport. Active transport is the transport of substances from their lower concentration to higher concentration by the expenditure of energy. Some other substances are transported passively. It is known as passive transport. Passive transport means free movement of molecules without the requirement of energy ATP. Passively, it is same as that of diffusion. Okay. Now let us come to the topic turgidity and flaccidity or plasmolysis. Then what is meant by turgidity? Turgidity is the state of a cell. When a cell reaches a state in which it cannot accommodate any more water, that is, the cell is fully distended, and that state is known as turgidity, and the condition is said to be turgidity, and the state is said to be turgid cell. The cell is said to be in a turgid state. So here, when does it occur? When the cell is continuously getting solution where the concentration of solute molecule is very less, or a lower concentrated solution like the water. If the cell is in contact with the water, so endosmosis takes place and the cell reaches a state known as turgid state, where the cell cannot accommodate any more water. So let us understand the turgidity or the turgid state of a cell with the help of a diagram. Here you can see a turgid cell. Vacuole is enlarged and the cell content is fully distended. And during this time, the turgid cell a pressure is exerted by the cell content towards the cell wall, and this pressure is said to be Turgor pressure. At the same time, cell wall is also distended and the cell wall exerts a pressure towards the cell content, and that is known as wall pressure. So in this condition, cell wall and cell content fully distended. So the Turgor pressure counterbalances the wall pressure. So there is no osmosis. That is why in this condition the cell cannot accommodate any more water, even though water is available to the cell. And this condition is said to be turgidity. State of the cell is turgid state. The next one is plasmolysis and flaccidity. So plasmolysis or flaccidity is the opposite of this condition, opposite of turgid condition. Here what happens in plasmolysis, let us understand, or in flaccidity what happens, let us see. This diagram shows 4 different stages of a cell. Here C, D, E and F. C is a turgid cell. D, E and F is the condition of the cell after immersed in a hypertonic solution, showing the successive stages of the shrinkage. So here in this turgid cell you can see the cell wall and cell membrane, it is intact, whereas when it is kept in hypertonic solution, so in hypertonic solution means the solution which surrounds the cell is with less concentration of water, or solute concentration is more. So water move out of the cell. That causes withdrawal of cell membrane or plasma membrane from the cell wall. And here in this diagram you can see very clearly the detachment of the cell membrane from the cell wall, and also shrinkage of protoplasm. Vacuole is reduced, size of the vacuole is reduced, and here you can see the full protoplasm is fully shrunken. Here you can see the cell membrane is also detached from the cell wall. So the withdrawal of cell membrane from the cell wall and the shrinkage of protoplasm when a cell is placed in hypertonic solution, because of the outward diffusion of water from the cell, causes flaccidity, or it is the cell is said to be plasmalized cell or a flaccid cell. So in flaccidity or plasmolysis what happens? Water move out of the cell, which results in the shrinkage of protoplasm and withdrawal of cell membrane from the cell wall. Okay. That means flaccidity is the reverse of turgidity. In turgidity, the cell is fully filled with water, whereas in flaccidity, water is lost from the cell. So a plasmalized cell can regain its original state by keeping this plasmalized cell in a hypotonic solution or in water. So when we keep a plasmolysed cell in water, it regain its original size, or it become again turgid, and that condition is known as deplasmolysis. Recovery or the reversal of plasmolysis is known as deplasmolysis. Then the terms once again let us understand: turgidity, plasmolysis and flaccidity. Turgidity is the state in which the cell is fully distended and cell wall is stretched because of the absorption of water. Plasmolysis is the contraction of cytoplasm or protoplasm from the cell wall. It is due to the withdrawal of water from the cell when it is placed in a hypertonic solution. And flaccidity is the condition in which the cell content is shrunken and the cell is no more tight. That state is said to be flaccid state. Then next to this we have the topics like uses of turgidity, and some other topics are also there. That one will be continuing in the next video. Okay.